IND, NDA, VLA, or other such documents. The next question is regarding retesting and when is retesting appropriate. The guidance document states that when conflicting results occur within a test run, that the user should consult USP Chapter 85 self-pilot limit test interpretation for guidance on repeat testing. As per Chapter 85, if the test failure occurred at less than the maximum valid dilution, the test should be repeated using a greater dilution, not exceeding the maximum valid dilution. Okay. The user should record the failure of the lab results, and all testing procedures should be specified in advance in written SOPs approved by the firm's quality unit. The next question is regarding sampling, storage, and handling, and that is sampling, storage, and handling important. The guidance document says yes, the ability to detect endotoxins can be affected by storage and handling. Firms should establish procedures for storing and handling, and then parenthetically, which includes product mixing, samples for endotoxin analysis using laboratory data that demonstrates the stability of the assayable endotoxin content. The document goes on to state that procedures should now always be validated to assure that endotoxin content is not lost during storage, and that if frozen storage is required, consideration must be given to the effects of previous law on endotoxin recovery. The previous law was that samples for endotoxin testing could be stored at 2 to 8 degrees for up to 24 hours before testing, and that the sample should be frozen if the sample could not be tested within 24 hours. The FDA has changed their opinion of that practice since the original document. Sample storage procedures should now always be validated to assure that endotoxin content is not lost during storage. This can be accomplished by spiking a sample with a known quantity of endotoxin and assay the sample at defined intervals after storage at that temperature. For example, a sample of a drug product could be spiked at 1 EU per mil and placed at 2 to 8 degrees C. The sample would then be assayed every 2 or 3 hours to show that the endotoxin content does not drop off. If frozen storage is required, the consideration must be given for the effects of freeze thaw on endotoxin recovery. For example, a sample would be inoculated at 1 EU per mil, vortexed, and then aliquoted into separate vials, which are then frozen at specific time intervals. An aliquot block would be thawed and assayed, and this would continue for the length of time the firm wishes to store the sample frozen. The next question was regarding pooling of samples. Can finished product samples for analysis of bacterial endotoxin be pooled into a composite sample prior to analysis? And the guidance document says yes. Pooling would generally be accepted for small volume parenterals of 100 milliliters or less, as long as the maximum valid dilution is adjusted to a proportional lower value because of the potential for diluting a unit containing harmful levels of endotoxin with other units containing lower endotoxin levels. FDA suggested in this example that if you pool 3 samples, then you divide the maximum valid dilution by 3. The document states that pools should be aliquots made up after 30 seconds of vigorous mixing from each of the product containers, and that the individual containers should be retained for possible retesting in the event of an out-of-specification result. Finished medical devices may also be pooled into a composite sample and assayed, and the testing for medical devices should be conducted using enriching, loading, and sampling techniques described in ISO 10993-1 and ISO 10993-12. The guidance document goes on to state that sampling may be adjusted for special situations. The pool must be maintained at the appropriate conditions to promote stability, and some product types should not be pooled. Examples of such would be products with an initial low maximum valid dilution, products manufactured as a suspension, and in-process samples from different in-process stages. The appropriate storage conditions must be determined on a product-by-product basis, as was discussed under sample storage. For example, for an initial low maximum valid dilution, if pooling 3 samples and the MVD is 1 to 3, you might be testing right at the MVD. Products manufactured as a suspension may have sample homogeneity issues, which may present significant interference issues, and it will be extremely difficult to ensure the homogeneity of the samples. The next question is about alternative assays. May a firm use alternative assays to those in the USP for a potential article? And the answer to that from the guidance document is yes. Firms may use alternative methods and/or procedures if they provide advantages in terms of accuracy, selectivity, or adaptability to automation or computerized data reduction, and in other special circumstances. Such alternative tests should be validated as described in the USP General Chapter 1225, Validation of Potential Methods, and be shown to achieve equivalent or better results. Better validation should be in accordance with the requirements of USP 85 and of USP Chapter 1225. That document goes on to state that recombinant or sugar factor C assays may also be used. An example such as pyrogene. Bruce Loverock, Barry Simon, Alan Baines, and Al Mergenson have already published the validation of 10 different representative pharmaceutical products in the January-February 2010 issue of Pharmaceutical Forum, from the online Pharmacopeia Forum of the United States Pharmacopeia. There is a one-time registration fee to get access to this free online reference document. The next question is regarding transitioning from one alternative DET method to another. And the question is, what is the best process for transitioning from one alternative DET method to another? This is done by comparing the two steps to verify the equivalence of the new method using USP Chapter 1225. For drug, animal drug, and biological products, the transition to a new method should be submitted in a prior approval supplement, or PAS, or by using a comparability protocol, CP, before using the new method for release. For medical devices, a 30-day notice may be appropriate for changes to quality control testing for all DET testing. This addresses changing from one alternative test, for example, recombinant factor C, to the NAT test, to another, but does not mean changing from one compendial method to another. However, if going from a compendial method to an alternative assay, the firm must first perform the validation and then submit to FDA in the form of a pre-approval submission. Once the firm develops a history with the alternative test, they may then submit a comparability protocol. The CP is simply an outline of what testing will be done, what parameters must be met each time before the product will be routinely tested with the alternative test. Once the CP is approved, the product submission for each product after that is reduced from a PAS to a CPE30, where changes being effective in 30 days. A CPE30 allows the firm to release the product into interstate commerce 30 days after the FDA receives the submission and not have to wait until FDA approves the submission. This could result in a time saving of several months per product. The next question is regarding Appendix E of the 1987 guidance document. So the question is, what happens to the endotoxin limit table in Appendix E of the 1987 guidance? The endotoxin limit table is out of date due to increases in the number of dosages and drug strengths since publication of the 1987 guidance document. Therefore, the customer or drug sponsor should use the calculation methods provided in USP or ANI documents. Monograph limits may not account for current product strengths or dosage regimes, and endotoxin limits should be checked using the calculations recommended in the standards. The FDA will no longer allow submissions to contain references to the endotoxin limits found in Appendix E. The FDA wants to see the actual calculated limits based on kilogram, including how the calculation was done for each product. You must show the math in the submission. Limits for multi-component products should be calculated based on the expected contribution of endotoxin from each component. For example, with pre-filled glass syringes, the contribution of the syringe must also be taken into account. For products that would be mixed prior to administration, the content of the mixed product may not be able to be tested. Therefore, the content will be determined by adding the content of each of the components. The sum of the components must be below the limit of the firm established for the combined product. If the single product limits specifications have not changed, ophthalmic products are a new category in endotoxin testing due to endotoxin issues causing health issues after being administered during intraocular surgery. The next area addressed in the guidance document is quality by design concepts and endotoxin limits. The document states that strategy should be based on product and process understanding in combination with risk management for consistent final product quality. It states that in-process testing should be used to evaluate the production process areas at risk of endotoxin inflammation or incursion. Furthermore, finished product release specifications should be considered when determining in-process limits for each phase of manufacturing. In quotes, qualitative testing may be preferable to limit testing to facilitate product quality trending and to identify and correct excursions before they exceed the specifications and cause product failure. These are the same ideas regarding in-process testing and raw material testing that were promoted for over 20 years, and actually much longer. The firm should assess the quality of their incoming raw materials and assess the relative contribution of each raw material in the final product. Animal or plant materials may have greater contribution of endotoxin than a synthetic inorganic material. The raw materials will be treasured over time to assess their suppliers' quality systems. Trending also allows manufacturers to not use a raw material if it exceeded the limit set for that raw material to prevent the final finished product from failing the release assay. This is just standard process control. The next area that the guidance document discusses is the rapid pyrogen test. The question is, when is the USP Chapter 151 pyrogenicity test or the RADD test appropriate? The document states that for certain biological products, 21 CFR 610.13d requires a rapid pyrogen test. However, this test may be waived if a method equal to the rapid pyrogen test is demonstrated in accordance to 21 CFR 610.9. For human and animal drugs, some USP monographs still require a rapid pyrogen test. The user may substitute an endotoxin test or alternative cell-based assay if the firm can demonstrate equivalent pyrogen detection. Testing is performed in a side-by-side comparison of the rapid pyrogen test at various concentrations of the test article versus testing of the same solutions using LEL or an alternative test such as Lonza's Pyrogene. The rapid pyrogen test may also be required endotoxin and will still cause a pyrogenic reaction if found in the product. The next area for discussion in the guidance document are limits for veterinary products that targets multiple species. And the question is, how would an appropriate endotoxin limit be determined for a veterinary product that targets multiple species? And the document states that for use in multiple species, the limit should be based on the maximum product dose used on the smallest species. For use on juvenile and adult animals, the juvenile is usually considered the worst case. And if the weight of the animal is required to calculate the dose, you should use an average weight for that species. And an example of where those weights could be found is in the waivers of in vivo demonstration of bioequivalence of animal drugs, insoluble powder, oral dosage form products, and by-date medicated articles. The next area for discussion is limits for medical devices. And the user should refer to USP Chapter 85 and USP 161 for information. The user should also refer to the FDA guidance for industry document for full clarification. Limits for devices are found in USP Chapter 161, transfusion and infusion assemblies, and similar medical devices. The endotoxin limit depends on the route of administration, intended use of the device, and what the device contacts, for example, blood or the cardiovascular system, lymphatic system, cerebral spinal fluid, intrathecal routes of administration, permanently implanted devices, and devices implanted subcutaneously. The FDA added information regarding flushing the device for catheters where the internal surface is labeled as non-pyrogenic. Disassembly or cutting the device into pieces for devices that are too large to be adequately rinsed, and also immersion for implantable devices. For some examples of limits of medical devices, we find that the device limit stayed the same as which was listed in the AAMI guidance document, and that's 20 EUs per device. An example would be a device that contacts the cardiovascular system or lymphatic system, and the extraction volume would be 40 milliliters of non-pyrogenic water, such as water for injection or LAL reagent water, and testing 10 devices. And the concentration limit would be, in this example, 0.5 EUs per ml. If the device contacted cerebral spinal fluid, the limit would be 2.15 EUs per device, and again, using the 40 ml extraction volume, the limit would be 0.06 EUs per ml. Intraocular devices is a new category stemming from use of solutions which were not originally meant for use internally during eye surgery, which caused problems attributed to endotoxin contamination. The new guidance states that a lower limit may apply, but the firm could contact the appropriate review division for specific recommendations. This means that the limits will be evaluated by reviewers on a case-by-case basis, and a rogue formula may not be appropriate. The next area of discussion is screening for therapeutic drug products, and the question is, what is the FDA's expectation for regular screening of therapeutic drug products? The document states that the undiluted product should be screened, and that if ingredients interfere with the LAL test, the user should pollute to overcome the interference. And that the calculated MPD is the pollution of the sample at which the endotoxin limit would be detected, but it should not be the regular testing pollution. If interference is encountered during the product development, the firm should determine the lowest product pollution that would neutralize the interfering condition, and this is known as the minimal inhibitory concentration, or MIC. Finally, the guidance document goes on to discuss controlled standard endotoxins and their use, and the question is, are controlled standard endotoxins, or CSEs, still acceptable for use in running BT assays? Controlled standard endotoxins are endotoxin preparations traceable in their calibration to international reference endotoxin standards, such as those found in the United States Pharmacopoeia, or EP. They're manufactured and certified by an LAL manufacturer for use with a specific lot of reagent under defined assay conditions. CSEs have become an accepted source for preparation of standard curve calibrators and assay controls. They have provided cost savings to LAL users and help preserve the inventory of primary standards. Furthermore, the FDA encourages continued use of controlled standard endotoxins. Finally, I'd like to thank you for your attention during this presentation, and if you have any questions, you can contact me at the information you see on your screen, or you can also contact your sales representative or Lonza Scientific Support. Thank you very much.
Good morning, everybody, and welcome to today's webinar about LR. Thank you for joining us. My name is Ingo Giorgotti, and I'm an executive support specialist at Lonza. I'll be the moderator for today's webinar, and I would like to welcome our speaker, Dr. Kevin Williams. With more than 30 years of hands-on experience in the area of pharmaceutical quality control, Kevin is recognized expert in the field of endotoxin detection and is author of the reference book Endotoxin, Pyrogens, Testing and Deprogenation. In addition to publishing papers in leading industry journals and other forms of media, he is a briefly speaker at the Parental Drug Association and other related industry events. And with that, I will put you Kevin.
Thank you, Ingo, and welcome. Thank you for joining the webinar this morning. And I might add that this week has been the PDA conference in Bethesda, so we have been at this conference at the same time as the webinar, and during the presentation I'll probably make references to updated insights from both regulatory authorities and users at the meeting. So I'd like to speak here on low endotoxin recovery and provide some context and resolution from a broad biologics test perspective The general categories to be covered include what is low endotoxin recovery, some background and current regulatory status, how to recognize LER in products and processes, including whole-time studies, and an emerging view coming from the FDA laboratory centering around endotoxin as an innate immune response modulating impurity, which is a new view that's just now beginning to be looked at in terms of endotoxin detection. So the first section, what is low endotoxin recovery? It's a controversial topic, I would say even a polarizing topic. At the conference yesterday, they have a new little handheld gadget for the audience where you can participate in the discussion. They ask questions about people's various beliefs on LER, and it was humorous to the audience when the immediate answers go up and the answers are split basically 50-50 between the participants. So this is a controversial topic and has been circulating throughout the endotoxin detection community over the last 3 years. It was brought to regulators' attention by Dr. Joseph Chin of Genentech in 2012. In theory, LER is a masking effect manifested in the biophysical formation of a complex that blocks the ability of factor C, a component in LEL detection, of course, recombinant factor C detection, to bind endotoxin. LER subjected samples show poor MAT and rabbit pyrogen test recovery in mammalian-based tests as well. The common drug excipient polysorbate, when combined with a chelator such as citrate or phosphate, has been identified as the cause of the masking effect, commonly referred to as LER. Polysorbate is said to be used in more than 70% of protein formulations, so it's going to be quite a common issue. There's also some evidence, and the FDA reiterated this yesterday, that in some cases they've seen histidine contributing to formulation, low spike recovery of undiluted spike. This is not too surprising, as histidine is an amino acid that is known to bind endotoxin and is used in some cases to remove endotoxin in process manufacturing. Also, LER is considered to be distinct from what we refer to as common interference, just where the recovery of the spike is less than optimal. The difference being interference occurs in a dilution of the product, whereas LER occurs from spike into undiluted product, and also has a factor that includes the passing of time. So the current regulatory stance that the FDA has is that manufacturers who are submitting BLAs for market approval should include a stability screening requirement. These hold time test studies, and this is described in the 2012 Q&A guidance for industry on pyrogen and endotoxin testing. She originally pointed out this phenomenon before the Q&A guidance came along, but these type of hold studies are where users now commonly see the LER phenomenon. The guidance recommends industry performs these hold time studies to verify the amount of time a sample can be reasonably stored prior to final release testing. Why is the LER focus primarily associated with biologics? Regulators require a biological license application, a BLA, to be submitted for each new biological drug product before it's released to market. So the BLA is a formal request for permission to introduce a biologic product into interstate commerce regulated under 21 CFR 601 to 680. If the information provided in that package meets the FDA's requirements, the application is approved and a license is issued, allowing the firm to market the product. What's typically happening now is the BLA is approved, but the FDA is currently requesting follow-up studies post-approval for drugs exhibiting LER. Another difference between biologics and small molecule drugs or non-biologics is that for biologics, the rabbit pyrogen test has always been a requirement and still a requirement. So this pyrogen test has to be performed usually at the development stage, and then the BET, after having shown equivalence, may be used. So this is different from non-biologics, where equivalence between the BET and the rabbit pyrogen is assumed, so the rabbit pyrogen test does not have to be performed for non-biologic drugs. The requirement, as I said, may be waived if the method equivalent to the rabbit pyrogen test is demonstrated according to the Code of Federal Regulations, which states that it requires the applicant to provide evidence demonstrating that the modification will provide assurances, the modification being a non-rabbit pyrogen test, so that would be even the BET test. This has been somewhat of an assumption that it will automatically be substituted, the BET for the rabbit pyrogen test over the years, but now this automatic assumption, of course, is in doubt due to the LER issue, which the FDA is now requiring the interim testing of the rabbit pyrogen test until some of these issues are resolved, at least in the United States. How to recognize LER in your process, in your product, and we'll talk a little bit about these whole-time studies. This slide doesn't differentiate. It's a quick screen to see whether your product is exhibiting such a phenomenon from a more complicated and more formalized whole-time study. So the basic idea here is to directly spike into undiluted drug product or drug substance or in-process solution at the level of the specification or slightly below the specification. And then once the undiluted drug product is spiked, the product is held before testing it according to predetermined parameters, including the length of the hold, the sampling number and times, the temperature, the container type, etc. So a more formalized study for BLA submission would have all these parameters addressed, whereas if you were just starting in development and want to know whether you see LER, it could be a much less rigorous process. Similarly performed, but perhaps not down to the container types, etc. So the recovery of the undiluted spike into drug product, the percent recovery as compared to the theoretical amount will determine whether you're seeing LER or not. So typically these results can drop off very quickly depending on the exact formulation. So in a matter of an hour or two, you can see the complete disappearance of your spike, or it may trend downwards over several days. So a simple screening test such as this using control standard endotoxin can demonstrate the presence or absence of LER for a given drug product or other solution. Typically the trigger for this kind of question during the BLA submission stage would be, does the product contain polysorbate? Without polysorbate, LER, of course, I don't believe has been seen or would be referred to as a more typical inhibition that could be overcome by traditional means. So whole-time studies are being requested for all formulations containing polysorbate surfactants. And of course, these studies include the spike undiluted drug product with CSE, and the CSE is still being recommended by the FDA as of yesterday's meetings. They are not ready to get into the variability associated with naturally occurring endotoxins at this time, and it's thought that the CSE will represent a worst-case scenario for such studies. And they were also advocating as I said, the inclusion of the container type, storage conditions, reasonable sample hold times. So these parameters would take into consideration your knowledge of your own specific process. So you really want to see whether the phenomenon is occurring as per how your own process is performed. There's been some question over time, and there still is some question and some vigorous discussion about whether what would be more appropriate to use, a purified standard such as control standard endotoxin or a so-called naturally occurring endotoxin in these whole-time studies. And thus far, the regulators, at least here in the U.S., have stuck to their contention that the use of NOEs is not going to be desired at this time. So BLA is using both CSE and NOE preparations have displayed conflicting results between the LAL and the rapid pyrogen test. And as we've seen in the previous slides, this is the equivalence that the FDA is looking for by law. So Dr. Patricia Hughes yesterday showed a similar slide to this, which really just points to all the different answers that the FDA is seeing from various BLA submissions that they receive. So where they can really see what's coming into some significant degree of detail, and they have seen these contradictions in recovery based on whether there's LER or no LER. The red recovery in the middle is the worst case scenario, where LER has actually caused you to miss a contaminant in your sample. However, that sample remains pyrogenic. And the regulators have spoken to the confounding results. And this is due to a variety of formulations of different proteins, different excipients, study designs, controls, test methods, standards, and reagents used. So you can see here in this case study that the LPS spike or purified CSE spike on the left side exhibited LER within 24 hours, and but it also did not show a pyrogenic reaction in rabbits. So the NOE was used on the right side, and it did solve the problem of LER quite well up to 23 days, but it was non-pyrogenic in rabbits, so you can't really have a standard that doesn't fulfill the need to give a positive result when it's present and a negative result when it's absent. So NOE data from rabbits is inconsistent with the results of the LAL method. Here's an example I found, and this is from February 2014, so it's possible it could already be outdated, although it still seems consistent with what the FDA is saying today. It shows their expectations in performing such an LER study. So this is concerning the BLA post-approval commitment that the company makes to the FDA. So they submitted their LER data, but the FDA comes back and says, however, the studies were conducted with only one drug product lot and with one endotoxin spike level. The goal of the study will be to verify that the method reliability with 3 additional drug product lots using the spike concentration is closer to the acceptance criteria, and then some of the information is blocked out that's specific to the drug. So from this small excerpt, we can see what the FDA is recommending for these whole time studies, and they want to see test data for at least 3 different lots of drug. They want those spiked close to the drug specification at or below. They want to see the test length of test out to 8 days. To me, I take this to be day 0 is really day 1, so out to 8 days is a 7-day test. And finally, they are still advocating the use of control standard endotoxin as a worst case. If we ask ourselves, what exactly is the worry with LER in this biologics manufacturing process? Of course, there's parts of the process that historically has to be gauged using LAL, and this is the transient occurrence of microbial contamination. So if you have a process that's going along and microbial growth occurs, gram-negative organisms, which are waterborne organisms, then these can grow up and then subsequently be killed by the process. But it's the residual that's of concern here, both in terms of the pyrogenic reaction and, of course, the effect of microbes, the concerns of alteration of the product properties. Some industry participants seem to hinge the LER concern on the biological activity of the aggregated versus disaggregated LPS, be it standard or natural endotoxin. So it's easy to see that using LAL, that LER solutions are disaggregated and become inactive. But this is not so straightforward to see in mammalian systems, and I wanted to speak briefly to that. Endotoxin normally exists in an aggregated state, so this theoretical disaggregation of the aggregates to monomers is pretty well agreed upon as the basis for the LER phenomenon. So I mentioned that straightforward in the horseshoe crab-based test that the disaggregation of endotoxin to monomers is what is preventing the recovery of a spike. But on the other hand, in mammalian systems, we see that some of the BLA submissions user data show that even though these solutions are subjected to LER, presumably to monomeric endotoxin, we see that in some cases they're still pyrogenic. We have the confounding fact that many biologic infusions are inherently pyrogenic in terms of the way they're dosed. For example, monoclonal antibodies are pre-dosed with a steroid and anti-fever drugs. So this is a typical reaction with in a clinical setting to include fever. So you can't really say there's no fever in today's modern drug treatment, although the cause of that fever is still, of course, under debate. And finally, lipid is a well-established endotoxic principle of LPS, and it fits into the human toll-like receptor 4 architecture as a single molecule, not as an aggregate. So in this basic question, we have, do monomers matter? And that would be in the mammalian system. Dr. Jack Levin, who spoke a year ago at the PDA, he showed increasing LAL gelation time with increasing disaggregation by hemoglobin. So in the mammalian system, it was seen that hemoglobin actually resulted in a greater detection, not a lessening of detection in mammalian systems. And there are also more than 200 acute phase proteins in mammals responding to endotoxin. One study that's often cited is the Mueller study, that only the aggregated forms of endotoxin are biologically active. But if you look at this study, they say it was performed in serum-free conditions, so